that I wanted to stay away. So these are the ones I said I didn't stick with first. So the clear cotton.
Yes.
And like sometimes I get too, and then, but I'm red because.
A lot of mine are red.
Oh, really? Okay, so I stayed away from it because the one that we did have, it was.
Oh, no,
Okay, just wanted to make sure.
I think I probably have like 4 times.
Okay, so let me show you.
Most of mine are like Sonora.
Okay, so this one.
That's out, well, yes, yes, that's out, this. This is what we're just looking for. Is that, it is that.
I mean, I guess they're not always.
They're not.
Okay, good, yeah,
Because
okay.
they were picking, the way they're picking the taxes and stuff like that.
Okay,
So it came out a little different.
Okay.
But, so this one, we're gonna have to make that up. Because this one actually has an absolute plus, plus or negative.
Your what? Okay, cool.
And then this one, I'm just gonna choose that one.
She's like,
Because I just said.
she wanted one adjustment, so I was working on the lab and come to find out, I had actually messed up on one of my.
So I ended up having
For that
three
test,
people
so the results from there, and then we're able to positive.
Oh,
Right.
yeah,
But.
I would.
But I could also use the phenol red, but I'm like, I don't want to.
Yeah, there's some tests that I'm trying to, have one that's okay for us. Her fatigue, that one, she does have it, but I'm trying to avoid that one because like it didn't work the first time, so why am I trying to avoid it?
We also had a weird one, when the plates,
Oh, the, MSA. Mannitol salts. Oh, everybody's did. So like, there was, she said that there was,
You know what I'm saying? Like, I feel like I can get different.
something that's not supposed to grow on it, but it did grow on it, and it happened to everybody. So like, yeah, it happened to everybody, because she was like, yeah, that's not normal.
Yeah, I know.
I was like, oh.
I always do HIV tests on
Yeah.
people to see, like, hey, is this something that we do it?
Yeah, I did too, I double checked
It
it
was
too.
like, it should not grow.
Should have been that, so I put that in my book, I was like, that's not supposed to grow.
Yeah, that's the only one I put. I was like,
It should not.
As expected, I was like, no.
No, it did not. Actually, because that was not supposed to go. How did you? Good. I got 100.
Girl,
Yeah, I was shocked.
I got a 93, but
That's still pretty
I
good.
already knew.
Like, I just
Which one was it? Which one's you?
So this started, mediums, like, explaining like where they are. So like.
Oh, yeah, yeah,
Totally.
yeah,
And
I
I was
know.
like,
I was, that one took me a minute. Like, yeah, I don't even know how I wrote it down.
Well, I mean, I got Jess, you should have like known what is the differential, or not differential, it's those different.
Like, so this,
It was talking about like,
I know.
I know it now.
Yeah.
But it's like, are there,
Do my voice.
like the media,
Yes.
it doesn't have like nutrients to feed it.
Yeah.
It just has like one.
Yeah.
Or it's like, I don't know, now that I have to go back and look at it again,
Yeah.
because it totally bothers me.
Yeah.
I left that, gosh, I know that I missed this.
Yeah.
And I don't know what I missed.
That's not really good though. I know, girl, I was like, oh,
Did she talk about the average on that?
yeah.
I made damn sure to make sure that I had like enzymes, substrates.
I had all that stuff. Like I made sure like I was even underlying some of the areas so I could like look at it really fast.
Yeah.
And, like, oh, there it is.
Oh, yeah, I know.
There's the answer.
I think the most frustrating part was waiting on.
Oh, that. Yeah. Oh, that was too. Like it was like you didn't have a lot just to fiddle around with while you're waiting. Like,
Yeah,
at
and she's
all.
like, wait,
What other stuff?
well,
Well, there was.
you know, like, but I want to see it, like, I want to see the thing, and then I want to write the answer.
There was one though, you can almost write all of it. before
Yeah.
you got over it, but-
Yeah.
Oh, and the other one that I thought for sure I did not get was like one of the microscope ones where it was like a stain. Or what, like what
Yeah.
test was this,
Yes,
right?
yes, yeah.
And I was like, so I like literally had to look at it and then look at my photo of like what I took and I'm like, that,
Yeah.
like, honest to God, I don't even know if that was correct or not, but that was the closest thing. And I'm like, is that right?
Well, I don't know, they the same, we did, they have the same stuff.
It was, yeah. I was like, capsule stain maybe? Yeah, that's actually how I knew I was right. Because, like,
Okay,
I
yeah.
had to look through my notes and I'm like, there's only one that mentions background stain.
Yes,
Like,
yep.
there's none.
Like very specific.
Yeah, very specific, so that's how I like. Yes, exactly. Because the first one I looked at, I'm like, I don't know. It looks like a simple stain, it looks like a
Right?
gram stain, it looks like a capsule. It could be all of those, I don't know. One of those?
Well, it's funny because when I did my capsule stain, which is not really nice, but I used a different background stain because the red
Oh, the saffron, whatever it is.
no, not that one, the Congo red was it?
Congo red.
She said we could use another one, right? So I did, I used this one.
Did it temporarily move?
It did, and then, but my lab partner ended up getting, she had to redo hers.
Oh,
And
yeah.
then she did the Congo red background, and her picture was,
So good.
yeah, I was like, damn. So I took a picture of
Congo
hers.
red either. I think I might have used the same thing.
The first one?
Yeah, I mean, it was.
Now that I think of it, I
did
think
the
that was the
purple.
same, it was the same situation. Yeah, cause Liliana grabbed it,
Yeah.
like, we're using this, and I was like, oh, okay.
Okay.
Okay, whatever you want.
Yeah, we'll follow the directions.
Yeah, exactly.
Did you do anything exciting?
No, other than working on my TikTok.
Yeah, which
No.
I thought it was kind of fun.
It was kind of fun,
But
it's like a little like puzzle.
Yeah, but once I got started, I had to finish it because then I was like, oh, I forgot
Oh,
where I
yeah,
am.
exactly. I sat like after classes, I was straight to a party, and I sat there until I was done. Like pretty much done, like I hadn't done like the positive side or anything, but
Yeah.
I did the entire negative side, and
Yeah.
I got stuck, and I'm like, I don't know what I'm doing. So I'm like, let me go email her, so I told her, and she answered my question, I was like, okay. So then I redid it again, and then put it on that little share sheet, and like kind of figured out that one.
Yeah. What did you use for that? Just
Yeah, that's it. So make your little key on top, and just do use your initials, whatever it is, and just, it's so much faster, you have to just type it out the name.
That's good, because I was like, I
Seconds.
don't know.
Yeah, just do that, and then I'm trying to figure out if I do lines in between, or I'm just drawing lines.
Oh,
You know what I mean?
good call. You can totally
So,
draw lines.
yeah, so just do box,
Yeah.
and then draw lines.
Yeah, and honestly, I'll do it like you did. I'll
Yeah, just do little keys.
Yeah, because I was like writing all those down.
Super easy. Alright, what are you?
We are, I believe,
The virus one? This one?
yes.
I thought this one was
Welcome back, good to see you all. All right, so good to see you all. We have about 5 weeks left, so a lot to do in 5 weeks. So I did want to cover the pathogenic presentation, so we can talk a little bit about what that looks like. This morning we assigned the chapters for the groups, and then also set up the times, so we know the morning group will do the same thing in the afternoon when we get in there. So I just posted this, because I forgot to do it, of course, but I went ahead and under the content tab, content. There is a pathogenic organism presentation folder, and in here you're gonna find the presentation overview. This is what we're gonna talk about, and then this is the rubric that I literally sit there and grade you as you're doing your presentation. So this is what I will use during the presentations. So let's go ahead and talk a little bit about the presentation itself. So at the end of your textbook, there are 6 chapters. It is not 20 through 26. It is 19 through 24. And these each cover a system of the body. So respiratory, circulatory, neuro, genitourinary, so each one has a specific system of the body that it covers, and then it has organisms that affect that specific system. So I, at random, had a little pile of chapters this morning. You pick a chapter, you're assigned that chapter. That's the one for your table. So everyone is gonna work out of that one particular chapter. You are going to, as individuals, do a presentation, all right? So the presentation has to be on separate categories of microbes. So virus, bacteria, helminth, fungus, protozoan, and if you're doing neuro, you can do a prion, because it only affects the brain, right? So we can't do a prion in respiratory, it's not gonna affect respiratory. So these are the choices that you have. You don't have to do all of those, they just have to be different. So if you are a group of 4, you're gonna pick 4 different ones. So protozoan, virus, bacteria, and helminth maybe, all right, so that you cover. If you're a group of 3, then you're only gonna pick 3, right? So you don't have to do the ones, all of them. So you're gonna choose a disease to cover, and then you're going to do the presentation on that particular disease. So you can go to the chapter, see if you find something interesting in there that you would like to cover, you can do that. The only caveat is you cannot choose one that I have already talked about, okay? So you cannot do RSV, for instance. I'm going to be talking about RSV. That is off the table. I've already talked about cholera and tuberculosis. Those are off the table, right? So kind of look through the material that we've covered and make sure it's not one that I've already talked about, because you're gonna do the work and I'll go, whoopsies, can't do that one, all right? So you're gonna wanna make sure that I have not covered it in detail already. The fungus are tricky, so that's why I went real light on the fungus, right? So you can do most of the fungus, probably the only one is coccidioides, would be the only one, because I did a little more detail on that one. All right, questions so far? We good? All right, so these are happening on April 23rd, April 28th, and April 30th. So that's what we're doing in lab. We're not doing it in here, we're doing it in lab. Smaller group, try to make it as casual as possible. You can sit at your table. The pointer reaches all the way to the thing. You don't have to stand in front. If you'd prefer to stand in the back, you're welcome to do that. If you know you have severe anxiety around this, come talk to me. We will come up with a way for you not to have a terrible experience, right? We just want you to share your information, all right? So what does this look like? So this is 75 points in total that comes from this entire presentation. 10 of those points is a 5 to 7 minute introduction that you do as a group about the system itself. So if you're doing respiratory system, tell me about the function and the major structures, right? This is not anatomy and physiology, so you don't have to go deep into all the different tissue types, etc. But you do need to tell me, if you're gonna be talking about upper respiratory infections, what is the upper respiratory system, right? So you wanna make sure that you include that information. This is the group grade. You can choose one person to do the entire presentation. Just know your grade's gonna be based on that one person, so choose wisely. If you wanna split it up and each person does a part, you're welcome to do that. However you wanna handle it, you do that portion, okay? So far so good?
Yep.
The rest of it is individual, okay? So all the rest of the 65 points comes from your effort. So in here I have specific information I need you to cover in your presentation. So I need to know the name of the disease it causes, what are the symptoms, what is the typical outcome, do you die from this, what is the prevalence, what's the incidence of this, what is the organism classification, is it a eukaryote, prokaryote, is it a fungus, is it a helminth, etc. And then if you are the bacteria, I have specific things I want you to cover for me. If it's the virus, there's specific things I'm looking for. Eukaryote, specific things, and then prion, because prions are weird, they also have their specific characteristics as well. So obviously if I'm doing a virus, I don't have to include things that are covered in the bacteria, right? Does that make sense?
Mm-hmm.
Right, so those are the things I'm looking for. All right, below this, I've decided to leave out anything about taxonomy, so don't worry about the taxonomy, just leave that out. I should have taken it out. So then you're gonna talk about the mode of transmission. How do you get it? Is it fecal oral? Is it droplet? Is it fomite, indirect contact? So all of that information, how do you get exposed? Does it require a vector? If there's a reservoir, what is it? Are we the intermediate or definitive host if you're talking about a helminth or a protozoan, right? Is the sexual reproduction occurring in us, essentially? This is to get you started. You may need to not answer all of them, or you may need to add additional information. Mechanism of action. So this is specifically, how does it cause the symptoms of the disease? because it's gonna cause tissue damage. How does it get to the particular cells? Once it gets into the cells, what happens? Does it replicate inside? Remember, we talked about, you know, for Yersinia pestis, it can get inside of the macrophages, move to the lymph nodes, escape, and cause disease, right? So you wanna add any virulence factors, et cetera, in here, as well as explain what those virulence factors do in order to explain how the disease is caused. Then we want to talk about epidemiology. What is the incidence? What's the prevalence? What populations are at risk? How prevalent is the disease? And then control. So how do you control it? Is it controlling the vector? So you do mosquito netting. Is it particular types of antimicrobials that are specifically helpful? So you'll want to include that information. So this is 1, 2, 3, 4, 5 components. So these all need to be covered in your presentation. Your presentation also needs to be somewhere between 5 to 7 minutes. Timing is extremely important. I will cut you off, right? So make sure you've practiced, etc. So here are some other resources. I talked about PubMed this morning in class. I'll do that in lab this afternoon. The CDC, World Health Organization, they have some good. Medline may not be accessible anymore. I think I tried it this morning and it might be behind a paywall, which is a bummer. But a lot of the things are on PubMed as well. Pictures are very, yes, Janaya.
Sorry, just for clarification, so
Yeah.
the intro should be 5 to 7 minutes long?
Yes.
And then each person
Yes,
gets
5 to 7 minutes, yeah.
Okay,
So the intro is 5 to 7, and then each person gets between 5 to 7. So that's why we only do
30
2.
minutes.
Yeah, so it's about 30 minutes altogether, maybe 40, depending upon questions, et cetera.
Got it.
That's why we have to time and be careful, or we can get late. All right, so figures are very important. Pictures are very helpful. This is awful. See how much text I have on here? You cannot possibly read this, so lots and lots of text, too busy. Try to have less text. You're allowed to have cards. You can have it written down, whatever you need to do to help you to give the presentation. Just don't sit and read it. Like, it's a little bit boring to do that. One thing I do want to point out, because somebody is definitely going to do, necrotizing fasciitis, it's going to come. I can know it's going to happen. We are a microbiology class, but not everybody in here wants to be a nurse. So let's not horrify others with terrible pictures of people in body bags. I've had that happen before. So please don't do that. Please aim about PG-13. And if you know something's coming up that might be a little upsetting, kind of give a trigger warning. Be like, okay, this picture might be a little rough. If you don't want to look at a worm in somebody's eye, you can prevent that from happening, because somebody's going to talk about that one too. It's very interesting. Terrible, but very interesting, all right? Otherwise, the other thing that's important is that you give credit. So put the link of where you got the picture from, cdc.gov, et cetera. Otherwise, it looks like you took the picture, and that's a form of plagiarism, right? So you shouldn't be doing that. It's easy to organize if you just do it based on points 1, 2, 3, 4, and 5. I don't know if you've taken classes before, but typically it's a minute per slide. So when you have a slide up, you should be thinking about a minute per slide. So if you have 25 slides, what does that tell you?
Holy smokes.
It's gonna be too long, right? So you kind of have to, you're gonna have to cut this down a little bit. And make sure again that you are timing it. I will cut you off, make sure you know the material, that's part of your points from the rubric as well. Okay, so far so good?
Yep.
All right, we actually have a really good time with this. A lot of people, it's pretty fun. So one thing that you have to do 1 week prior to your presentation, 1 week prior, there is a drop box where you need to tell me exactly what you're going to say. I don't want to know, I'm going to talk about this. No, I want to know what are the things you're going to talk about. So give me a rough outline of each of those sections. That way you don't accidentally say it's a prokaryote when it's a eukaryote. We can fix some of that before it occurs, all right? Listen to me carefully. If you do not turn that outline in, you are not presenting. It is the end of the semester. I'm over it, all right? So if you're late, tough, it's over, right? So please do not be late on that outline. I will warn you and warn you and warn you. Kind of forces you to get it done, right, so that you're ready. Questions on that, right? So this part is worth 10 points right off the bat. You get it turned in. I will look at it as soon as they come in. I start looking at them just so you have the feedback as quickly as possible. Okay. All right. So also, you have to attend all 3 sessions. If you do not attend all 3 sessions, you will lose 5 points. So things come up. I get it. You know, kids get sick. You've got to go to an appointment. that's fine, just let me know ahead of time, and we'll kind of work through whether that's a good enough excuse. And then I may not take the 5 points off, right? So you need to attend all 3 to not lose those 5 points. Macula, I saw your hand, you good?
Okay, when I'm listening, let's say I'm listening to your lecture and you mentioned something about helmet, and I'm gonna talk about helmet.
Yeah.
I can't use that?
You can talk about helmets as long as you don't talk about schistosacrum, whatever it is I talk about,
Okay.
right? So today I'm gonna talk about taniasis. You can't talk about taniasis, right? So you can talk about a different one, no pinworms, etc. All right, so one other thing that we're gonna have happening is you are going to present or give to me 3 days. I'm not as picky about this, because if I'm honest, I'm doing it morning of. I'm pulling this stuff and making the things in the morning. So you are going to come up with 3 questions about your presentation. So true, false, multiple choice. True, false, multiple choice about your specific presentation. I take all those questions for the day, I put them together in one document, and I hand those out at the beginning of the class, and you gotta take that test while the people are presenting, right? So you have to pay attention. Look what I just did to you. The nice thing about that is it's 10% of your final grade on your last exam. So you get them all right, it's 10%, right? 10 points right there. So you wanna make sure you are paying attention. Get these, ideally 3 days before, there'll be another Dropbox. Please make sure it's in Word. Don't do something weird, because I have to pull them and put them into a single document. If you put them in something weird, I'm gonna have trouble getting to it, and I'll be cursing you as I'm trying to fight through it at 8 a.m., because that's what's gonna happen. You will not, if I don't get in by that time, you'll just lose the 5 points. So unfortunately, lose 5 points. You won't have presentation questions. Okay, so the last thing is participation. So over the course of the 3 days, you can earn up to 6-point bonus by asking questions. So just ask questions, right? I take notes of who asks questions, and just kind of, that's how we get kind of into it. It's pretty fun as we go along. So 6 points from that. It's okay if you don't know the answer to the question, just say, I don't know, and then everybody looks at me, usually whether I know it or not, I don't know, but usually people know. They do a lot of research, and they can have a lot of background in their heads about their organism, so don't be intimidated by it. It's okay if you just say, I don't know, that's okay, it's a good answer. All right, questions? All right, so our first presentation is the 23rd, which means, was that the 16th? April 16th is when those outlines will be due. So it's coming up in a couple weeks, right, to start thinking about it. And if you want to shoot me an email and ask if I'm going to do this organism, is this okay? Also make sure that organism actually affects your system. Don't pick something that doesn't affect your system. That will not work. Yes?
So you said you could do like 5 slides for like each section?
No. So usually it's a slide per section, maybe 2.
Okay.
So maybe 10. 10 is usually enough. If you do 25 slides, it's going to be too long. It's way too long. Yeah.
So like for the entire presentation, including like the system overview, bacteria?
So the system overview might be 2 or 3 slides. That's pretty typical, 3 to 4, depending.
But for the entire thing, you're looking for like roughly around 10 slides?
I don't have a number. Yeah, I just know when people do 25 slides, it's gonna be too long. I already know it's gonna be too long. So try to take a minute per slide. So 7 to 10 is probably a good number to aim for, right? All right, thoughts. We're gonna assign the chapters this afternoon, and you're gonna assign your date. So you'll know what date. I will post all that information for you for the message this week in case you forgot and you need to be reminded. I'll have that in the list for you. Yes, Lydia?
Do you want, as a group, our presentation to be together
No.
in one PowerPoint?
Yeah, so you don't have to put it all together in one PowerPoint.
And
If
how
you
are we
wanna
gonna
do
get
it in
it?
one PowerPoint, you're welcome to.
How do we get it to you?
You don't get it to me. Yeah, I have a Dropbox that I will put there, so you could put it in there, so just as a backup, but I would have multiple ways to access your PowerPoint. Yeah.
Cause I'm just wondering about putting it on the screen.
Yeah, so I'll have a Dropbox, so if you wanna put it in the Dropbox, you can, but I would have it on the old thumb drive, put it through email. Some people download it from Google. That causes a bottleneck, just to warn you, because everyone tends to do that. And then I've got 15 million email addresses trying to get off Gmail. Yeah, so if you're gonna do that, I would do it as a group, right? So do one as a group, so that way we only have 6 groups that are fighting access to the Gmail accounts. But we like to get in there and we get them set up. But I will have a Dropbox if you just wanna drop the PowerPoint in there, you can do do that as well, but I'm not looking for it actively. Okay?
We're gonna do it in one.
All right.
We should do it in one. Nobody's here.
Okay, any other questions on that? Good questions. All right, so one other bit of housekeeping, couple. So I'm not gonna be here on Thursday, so hopefully you saw that message. I'm going to a conference in Salt Lake City,
Yay.
right?
I'm sorry.
Yeah, it'll be fine, it'll be fine. So I'm not gonna be here. Yeah, at least it's close, it's like an hour and a half flight, it's not bad. Obviously, I had trouble finding somebody to come and teach, not a lot of people teach microbiology. So we are just gonna turn it into a lab work day, which is a great day for a lot of gram staining that you're gonna do. So my morning group will go from 8:30 to 10:30, my afternoon from 10:45 to 12:45. Carly Knudsen is gonna be hanging out with y'all. She's an animal biologist, she knows nothing about micro. So she's not gonna be able to help you other than pulling things out and making sure they're on the table and the gas is on, all right? And she's watching for chemicals around fire, and that's it. So lots of gram staining, that's why I gave the lab information sheet. So kind of follow that information sheet, gram staining. The goal is to get an isolated culture, hopefully, on Thursday. I'll warn you right now, this is the hardest part. This is the part, sometimes it takes a couple rounds to get the isolation done. So don't freak out if you don't get it. Just make more colonies and culture and just try it again. So if you get it done by, not even next week, but the next Tuesday, by the time you get to that point, it'll take you 2 days to finish the entire rest of the project. Yeah, it's super fast, because we do an OF glucose, and then you inoculate everything on your key.
Oh.
And then you just read the results in 1 day. It goes super, super fast.
Okay, let's see.
So again, I know it's not gonna help to tell you not to panic, because you'll panic. But this is the hard part, right? So hopefully you'll get it on Thursday, but if you don't, it's all right. We have more time. Questions? Yes, Cassie?
Can you just do it at the end of lab Thursday? on the
Not
7th?
this Thursday, because I'm not here.
Yeah, but this, or Tuesday, I'm sorry, the 7th.
Tuesday the 7th, yeah, I need your key Tuesday the 7th. You can pull the Bergey's book while you're in there and work on that. It'd be a great thing to finish up. Very important, just to clarify, the only thing I'm looking for is all the organisms on there. I'm gonna count the number of organisms that I see on your key and make sure I can follow them all the way through from the top to the bottom until they're all isolated. And I make sure that you have tests on there that we actually have. Whether or not it's correct, I'll figure that out. So hopefully you got that correct. So I need to have the organisms listed somehow. So you have to have the numbers or the initials so that I can count. Be like 1, 2, 3, 4, 5, 6, 7, 8 on this side, and there's 8 on this side. Okay, and then I can follow them. Because if you just give me the test, I can't. That doesn't help me. Yes, Lillian.
Just a note, the Bergey's, at least the one I saw in the lab, is 7th edition. The Learning Commons has 9th.
Oh, perfect. They have an updated one. Good to know. Yeah, they're pretty much similar. The only thing that's really changed is one of the names, Citrobacter diversus, I think it's changed its name, but it should be okay. All right. One more thing. Okay, there's a lot. I'm like, dang it. All right, so one last thing, because I'm not going to be here Thursday, I don't feel right about giving you a test on Tuesday. That seems a little messed up. So we're gonna move it to next Thursday, so it'll be a week from this Thursday. So Tuesday, I'm going to post for you after today, probably the entire virus lecture, just based on what time it is already. But that material, go over it before you come on Tuesday so we can kind of review and answer any questions that you have, bring your weekly reviews, questions, etc.
So homework 7 will also be done?
Everything will move to Thursday. So
And
the
the
Connect
Connects,
quizzes,
okay.
everything will move to that date. Okay. But we will start with the new material on Tuesday. We gotta talk about controls, isn't that crazy? It's like the last part of the whole semester. All right, I think that's it. Am I missing anything? I think we're good. All right. Okay.
These two classes hit in the same weekend.
Also want to remind you all on these lectures, you might have seen it, but if you actually download them, I don't know if anybody's done that, but I also have the lecture underneath in the notes section. So
Oh, I
if you need
didn't
to read
notice
it.
that.
Oh.
Yeah, so every single lecture I have has it
Okay.
also in the notes section. Yeah. I annotate them all, so. All right. Oh, it feels like a long time since we talked about this stuff. Yes, question, Megan? This is for eukaryotes, so it's 11, I believe, still. Yeah. All right, so last time we were together, we had just finished up talking about protozoans and the oddness that is protozoans. These are the single-celled, unicellular organisms. They're typically differentiated based on how they move. So do they have cilia? Do they have flagella? Do they have no movement possible? Do they use pseudopodia? And then we can also kind of differentiate them based on whether they go into a cystic phase or do they stay more as a trophozoite. So trophozoite is the vegetative, living its life, doing its dreams, and then the cyst is usually what happens when it has to go through the environment. So it's sort of like an endospore that allows it to survive in the environment. When we're trying to figure out what they are, oftentimes these are pretty large organisms, so doing different types of smears helps us to be able to see them. I don't know if you remember when we looked at the eukaryotes, there was a lot of poop smears in there. That's because there was Valentinium was in there, as well as some vaginal smears that showed us the Trichomonas, and then blood smears that had the different types of sleeping sickness organisms as well. So it's very common that when you're trying to diagnose these, you'll look at bodily fluids and samples. You can just smear and then stain and be able to identify them that way. Outside of that, you can also do antibody tests. So we'll talk a little bit about ELISAs when we talk about viruses, and then you can also do PCR. So you can look for the genetic material specific to them. These are some of the different ones you might be interested in doing. Acanthamoeba, this is the stuff of nightmares. This is what you get from your contact lenses. It can end up in your brain. So these are interesting ones if you're interested in doing some of these. This is a gastrointestinal disease. And then we have some others. This is a STI, or a sexually transmitted infection that you can get, and the trypanosomas causes the sleeping sicknesses and Chagas, which is pretty messed up. So these are interesting ones if you want to take a look at potentially as one of the diseases that you can present. All right, so we are now going to move away from protozoans, which was part of kingdom Protista, and we're going to talk about helminths. So these are members of kingdom Animalia. So we're all in domain Eukarya. We had kingdom Fungi, we had the kingdom Protista, which again is getting reorganized, and then we have kingdom Animalia. So these are animals. This makes them extremely difficult to treat. Why are they difficult to treat? What are we? Also animals. So we have a lot in common with these guys. So things that would affect them can also affect us. You're going to see it's tricky. It's very difficult to treat these types of infections. And there's a lot of conversation about kind of the demon you know. So if you have a helminth infection of some kind, they often do not cause any symptoms or signs, which is crazy to think about, but they don't. And so maybe it's better to leave it alone in a way, because the medication and the treatment can be harder, particularly if you're living in rural, areas of the world. It could be protecting you against other helminths, right? So other types of infections. So that conversation comes and goes often in the field. So we're going to be looking at two different kinds, flatworms and roundworms. So they're exactly what they sound like, flat. These are the flat tapeworm type structures. The flukes are in this as well. And then roundworms are round, long, thin nematodes. And I think of SpongeBob, dang nematodes. All right. Okay, so overall, these are big. They are not small. Pinworms can be millimeter size, so super tiny. But some of our helminths can be 30 meters in length. That's like 60 feet. These are huge, huge organisms. So they're obviously there, but their transition, so the cysts that are released or the eggs that are released into the environment are microscopic. You can't see them, and so the transmission cycle is why they're considered microscopic organisms. It's because we can't detect them in the environment with the naked eye. These are multicellular animals, so they're going to have organs and organ systems. Depending upon where they're located, these can be very rudimentary, or they can be very normal, like what we have. So if you have something like a planaria, which we're going to talk about planarias because they're interesting, they're a free-living organism you find in pond water. They don't cause any disease. They have very developed musculoskeletal systems, they have a nerve net that allows them to sense their environments, they're very good at digestion, etc. So they have all of the normal function. If I have a tapeworm living in my gut, what do you think is most important to them? I mean, they're in my colon. So the food is already digested. There's no reason to have a heavy-duty digestive system. I don't really need to go anywhere, so I don't really need a good musculoskeletal system, right? So those things are gonna be more primitive, where they are awesome at reproduction. It's disturbing how awesome they are at reproduction. A tapeworm can generate 250,000 eggs over the course of its lifetime, right? Or some of them within a day, they can do 10,000 eggs a day. So it's intensely horrible, the amount of organism reproduction that they can do. All right, so their life cycles are fertilized egg, that's the embryo. We have a larval stage, usually within some type of cyst-like structure, or that can occur in the digestive tract or some other portion of the body. And then the adult stage, which is the worm stage. And most helminths get their nutrients and reproduce in the host body. So remember again, we had two terms. We have the definitive host and the intermediate host. The definitive host, when we're talking about helminths, they're the one with the actual worm, because the worm is what's producing the eggs in the sperm, and it's typically producing the embryos. So if you see the worm in that individual, that is the definitive host, right? The intermediate host is going to be the ones that have intermediate stages, and they can have some complicated life cycles as well. All right, so overall, we can have different morphologies in terms of reproduction. So you can be either monoecious or dioecious. So you can kind of see mono and di in here. So monoecious is another word for hermaphroditic. That means that same organism has both testes and ovaries. So the entire organism is capable of self-fertilization typically and can create embryos from that pathway. Dioecious means that there's two different, male and female, right? So we have two different worms that then come into contact. It is pretty typical for the worms, however, the males to have spicules, which allows them to grip the female, right? So they're able to kind of hold on to the female so they're always together, or they'll have a cleft that the female sits in the entire life. So while they are two separate species, they're always together, right? So they're constantly fertilizing eggs as a result of that, right? So where do they come from? How do you get helminths? What do you think? What do they tell you about meat? Freaking
Raw
don't
meat.
undercook your meat, right? So food, contaminated food, fecal oral is very common. So water transmission and food, contaminated food and water are your most common mechanisms that we hear about most consistently. If you think about in parts of the world, they have to bathe, wash clothes, get their drinking water all from the same location, right? And you have animals and people, waste matter getting into that water all the time. And so this is putting them at exposed risk all the time for the transmission of these organisms. The other place that you can get it, which is a little bit freaky, is some of these can actually penetrate the skin directly. So walking around barefoot in particular areas puts you at risk, particularly if there's fecal material from animals or humans that puts you at risk for picking up organisms. I ran around all the time in bare feet as a kid. Common things, so hookworms, you can pick them up from that as well. And then some of them can be transmitted through mosquitoes and tick bites and different large organisms. So something like filariasis, so lymphatic, that's where you get buildup of worm in the lymph system. So it ends up creating the large limb, the blockage, and they end up with huge limbs. That is caused by a mosquito transmission of a helminth. Yeah, it's the same thing as the elephantitis. Yeah, it's filariasis. All right, so here are some different nematodes if you're interested in maybe talking about one. River blindness is very, very interesting. This is one of the ones where I'm like, watch your pictures, because you can see the worm in the eye actually moving around. Guinea worm, this actually comes from getting in through your feet. Typically, this is one that you pick up as well. So lots of interesting ones if you're interested. I'm gonna talk about Taenia and Schistosoma as today. All right, so let's talk a little bit about our platyhelminths. So platyhelminths are flatworms. These are the flat guys. So this is gonna include the trematodes and the cestodes. Trematodes are called flukes, right? They're not small, they're usually about, what is that, 3 centimeters or so in length. They're a decent size. They are typically monoecious, so they have reproductive structures that are all part of the same. Paragonymous is the one we're gonna talk about, schistosomiasis. I might leave for later in case somebody's doing the circulatory system. But this is a typical structure. So again, these are not small. You can see the ovary and the testes in the same species. They have these suckers that allow them to adhere to tissue. So if you think about a lung fluke is gonna be in your lung, and you have this constant kind of up and down, that's gonna be pulling on it. And some of these will end up on livers, they can end up in your bloodstream, et cetera. So they have to be able to maintain positioning. All right, so we're gonna talk just a little bit about Paragonymous. So this is a very significant problem in Asia, parts of Asia. And it actually is, again, helminths can have very complicated life cycles. This one actually requires 3 hosts. How in the world that ever happened, right? What the heck? A human, a snail, and some type of crayfish or lobster. So you have to have all 3 to complete the entire life cycle. We are the definitive host. We are the one that has the fluke, so the worm is in us. That is where the reproduction is occurring. So initially, this is a lung fluke, so it is going to end up in your lung eventually. Unfortunately, the way that it gets there is a little creepy because you ingest the eggs, the fertilized eggs, or the developing egg. This gets into your digestive system. It does a little bit of development in the digestive system and then penetrates into your bloodstream and then moves into the lung. So it moves through your bloodstream to the lung, so there's some chemical signaling that's telling you what tissues to go into, and then it attaches to your lung where it starts developing the eggs. Fertilized eggs get caught in the sputum, you cough it up, you swallow it, and then it becomes a fecal transmission. So it goes out that way through your body typically. This gets into water through the feces, and then these guys develop, and they actually will then burrow into a snail in the ocean. Once it's done that, it does some additional development, burrows out, and then gets itself into a crayfish. So how do you think we get it?
Eating.
We eat undercooked seafood, right? So if you eat undercooked seafood, you can pick up these developmental stages and start the cycle again, right? So again, cook your food. Be careful that it's cooked well so that you can avoid this in areas. All right, so that was creepy. Let's make it worse. So now we're gonna talk about cestodes. So these were the trematodes. These are still part of platyhelminths. Cestodes are the tapeworms. So basic structure, they have a scolex. So you see this thing up here? This is the scolex. It's the stuff of scary, scary nightmares. They have one at the Science Museum in DC that they've blown up. So it's like this big giant head, and you can see on it like the hooks. So it literally has hooks to help it attach to your intestine. Because you imagine peristalsis is happening, you have fluids going through this, sometimes you get diarrhea, et cetera. It could flush it out pretty easily. So these adhesion points allows it to stick in place. Once this is formed from the egg initially, then it starts creating these things called proglottids. So these proglottids, they are the tape, when we think about the tape. So these are the individual segments, and in here, notice it's just reproductive. So it's testes, ovaries, and a way for the fertilized eggs to get out, right? So when you actually have a bowel movement and you have one of these can come out in the fecal matter, right, and come into the environment. They have a cuticle that surrounds them that prevents them from dehydrating, so they can be in the environment for months. They can be out in the environment for months. And you come into contact with them by coming in contact with contaminated food, contaminated water, et cetera. Or you eat something that has come into contact with contaminated food or water. All right, so our example of one that is, we're the definitive host. So what does it mean on the definitive host? What have I got? I got the worm, right? So I've got the worm in my gut, all right? So I've got the scolex, it's producing the proglottids, these are getting lost into the environment. So these are the taenias. So either the pork tapeworm, the beef tapeworm. So even though it says pork and beef tapeworm, they don't get the worm, right? We get the worm from eating undercooked meat, right? So that's where it comes from. So this is the life cycle. This is from the CDC. So here is the different shape on the heads. You can see them a little bit different. So this is the beef tapeworm, this is the pork tapeworm. Same idea. Typically when you get a tapeworm, you only get one. You'll have one tapeworm. These are the ones that can grow 40, 50 feet in length. So they can be the entire length of your intestine, essentially, that you have in there. And they can live decades, 30 years at least. So they can be there a very, very, very long time. What's very strange is you would think with something so big that you would have symptoms. Most people have no idea. You usually have no symptoms at all that you have a tapeworm. So you could have one right now if you're like, uh-oh, and not know, right? So you just don't know. So something this large really doesn't tend to cause anybody any problems, but it can, because you can have these break off. If you're a younger person, children, older person, these can cause blockages, and it can cause some issue, but most people have no idea.
And it causes like anorexia.
So that is, yeah, that's like the urban legend that you could buy these and they would give you a tapeworm so you lose weight. I think there is some signs that you could have some nutritional deficiencies, but not usually, yeah. My mother had a friend, I have all these stories, don't I? Because my parents, my dad was a minister, grew up in this church, and so they had a lot of missionary friends. And so one of the women, she came back from, doing this missionary, she was there probably 25 years, and she was always a little bit sick, a little bit anemic. And eventually they did figure out she had a tapeworm. And she had probably had it a very, very long time, but it took about 10 years for anybody to kind of realize maybe that was the case from the travels that she had done. Because she looked like a little white lady, with her Nazarene hair. And why would you think she did anything like that? But, yeah. Is anemia common? I don't really know. Again, usually you don't know. You really don't have any symptoms or signs. Maybe you notice something in the fecal matter, like you kind of notice something might be coming out, and so it makes you a little like, what is that? But they're small, so it's not likely that you're going to notice, because how many people actually look? Nobody usually. Yes?
So because it doesn't cause any symptoms, can it be argued that they're more commensalism than parasitism?
Yeah, this could be a commensal relationship, potentially. This is the one I was talking about, because if you have this tapeworm, it prevents other infections, right? So it's already taking up the space, so maybe in certain areas of the world, this can be protective, right? It can be protective. Because the drugs to take to remove them are pretty intense, right? It does make you feel bad, etc. And the worm has to come out all the way, and all of these things. Again, that's kind of a hypothesis people play with, and they go, no, we need to treat it, and then they back up, and then they go towards it again, no, we need to treat it. So right now they're kind of going towards it again. Yes?
There was this trend where people were eating papaya seeds to try to get rid of parasites and things like that. Would that potentially?
I don't know. What is in papaya? Is it cyanide in papaya
Yeah, I think
seeds?
so.
Maybe it was
People
the
were
cyanide.
just eating whole papaya seeds and just eating the seeds to, yeah.
Wow. I don't know. Cyanide seems bad for us as well.
Yeah.
I was gonna say, wait a minute.
So good.
They have the same, their cellular respiration system is the same. So it makes sense. Some of the drugs that we'll talk about actually can target components of their cellular respiration that doesn't target ours. So maybe it's something like that. I'd have to look it up to see. Maybe I'll try to remember. That'd be interesting to know. Yes?
So, like a dewormer for a dog or a horse, 'cause we used to do that all the time.
Yeah.
So, when you're giving something like that to, let's say, horses.
Yeah.
Like, should you be careful of how many times you're giving that to them? 'Cause
I don't
is it
know, because I don't
equally
know
as
about
toxic?
veterinary medicine, but I don't know also if they have more problems with blockage, et cetera, so maybe it's more important to treat. Usually causes paralysis of the worm. So the worm becomes paralyzed, isn't able to hold on anymore, and then it just gets flushed through the system. So the vast majority of drugs that we'll see just causes worm paralysis. I think ivermectin does. What is the other one? Chromethrin, I think, is another one, also causes paralysis. It just has, something goes after their tubulin, and so they just can't hold on anymore.
That makes sense.
Yes.
How do wastewater treatment plants prevent that from spreading?
How do you prevent these from spreading? These are big enough you can filter, right? So filter, like when you do your life straws and all that, when you're filtering water, it's mainly to get rid of this kind of stuff, because these cysts are big enough you can filter with your
Because it
life straws.
replaces easily the segments, right?
These still would be big enough. What doesn't filter very well are large bacteria, viruses, some bacteria. but they definitely get rid of helmets. That's part of why they filter. And then they do treatments, chemical treatments. I mean, nobody does good against bleach typically, and chlorine-based stuff, so that's part of what we do in this country to prevent it. And then also trying to come up with cleaner water sources in other countries. That's why they'll do wells and try to set up situations where there's a community well that people can go to instead of going to the river, to try to prevent that contamination or lower the chances of contamination by doing that. Yeah. All right, so let's talk a little bit more about what happens. We get the worm, we're living our lives, doing our thing, and we end up getting some proglodids out into the environment. We have our cattle and our pigs coming in contact with these different species. They digest or take in the little proglodids, which are gonna be our little fertilized eggs. They go into their system. They don't have the system that's correct to actually hold on or maintain the scolex and the worm. But what happens to them is in their system, in their digestion, they create these things called oncospheres. And the oncospheres, again, can penetrate into circulation, and they end up all over the body. So they end up in the tissues, the brain, the liver, all over the place. And they actually will get into the tissue and create a little cyst inside of these. So these are called cysticerci, which is fun to say. Cysticerci, right? So cysticerci develop in the tissue, and as long as the organism stays in its little house, it doesn't cause the animal any problems, interestingly enough. Nothing actually happens. They can live that way for years, like 5 to 10 years. You can have these guys in there. Eventually they do die, and they will rupture out. And then once they rupture out of the cyst, the immune system goes, what the heck? And it will actually start to attack it and cause damage, right? So you can end up having harm when these actually rupture. So we get it again by eating undercooked meat. So by cooking your, I think it's 145 for pork, 165 for beef, I might have them backwards, that actually kills these. So it'll actually destroy these guys in here. That's why they warn you about being too rare on your beef. You're kind of putting yourself at potential risk. In this country, we have really good mechanisms in place to prevent this. In the first place, in our farming practices, et cetera, it's other parts of the world, right, where we don't have the same luxury, basically, to protect themselves. One of the things that can happen, taniasis, this is when you have the worm. Cysticercosis can occur. So we can sometimes have it happen where we develop cysticerci. So instead of making the worm, it can get into circulation, and you can end up with these in your brain, in your liver, in your lungs, etc. as well. So you can have these throughout your body and not know it. And you're okay as long as they don't rupture. As soon as they rupture, there was a article about a gentleman who got in a car accident, and he had come, I think, from India originally, and he was like a physician, like in this country, and then he just went crazy, like after the car accident. It was like he had brain trauma almost. But it turned out what it was is he had an infection. He had cirrhosis, and these had ruptured in the brain and caused mass inflammation. And so that created the change in his personality. So they were able to treat it, and he actually recovered because of it. Very interesting. Very unusual, very rare complication that can happen. Much more likely you're gonna get the worm, all right? Yes, question, Cassie?
Some people who eat like blue rare steaks or.
Blue rare steaks. God bless you.
Yeah, I had a guy come in, I work at Texas Roadhouse, but I had a guy come in, he orders a steak blue rare, and he was like extra blue rare.
Just almost
And I
mooing.
was like, right? And he's like, oh, like, all right, cool. And then his wife told me that he had a pretty massive tapeworm like 10 years ago.
I believe it.
And he's pretty certain he has another one. And he's like,
He's pretty
"I'll
certain
give
he
you
has
steak how I like it." And I
Oh,
was like, that's so nasty.
Oh my God.
But it's still like.
It's America, right?
Blue rare
So
steaks,
you want
like-
your tapeworm. Yeah, that's usually.
Are we supposed, like, should we be getting them more like medium well?
I think,
Or,
I don't
like,
know what the
does it matter?
temperatures are for rare. Does anybody cook steak on a regular?
I don't know.
I do
I'm
that.
not-
What is it? What is it for rare?
See if it's like.
Rare.
Yeah, do you know what the temp is for rare?
Oh,
I wanna say it's like.
like 2 minutes on each side.
On each side.
Pretty
much, yeah.
I'm pretty sure one
Yeah.
forty
Yeah,
rare,
that's
it's
about right.
142 to 144 mid-rare, it's 146.
So those are right on the edge of okay,
Okay.
I think. So I think the 145 is probably beef, and the 165 is pork, a little bit
Yeah, I
hotter.
think so.
Yeah, so that's, I mean, restaurants cannot legally give you something that's gonna give you a tapeworm.
Yeah.
They'll get sued.
125 to 130 for rare.
What is it?
125 to
So it
130.
is low. So it's a little bit lower than it should be, but
I know we tip ours at my restaurant around 130.
130.
Yeah,
And then I wonder, cause we're at altitude, you know, things, I don't know,
Oh.
are different at altitude, I don't know. It's a very rare occurrence, again, because of our practices with animals. We prevent quite a bit of it, but it's possible, you know, so. This is where I always like to, people love natural, but we can naturally get a tapeworm if we're not careful, right? So we need to be careful. All right, so let's talk about another one that is where humans are the intermediate host. So that means we don't get the worm, but we can get the cysts more commonly. And that is gonna be Echinococcus granulosus. So this is very similar to what we've seen with our other organism. So this is the life cycle for that guy. We are an intermediate host, but we're also a dead-end host. And I'll tell you why. This is a predator-prey relationship. So our sexual reproduction is occurring in the predators. So this, they're showing you a wolf here, but it could be a cat, you know, so some type of cougar or lion or, you know, some large predator animal. Your dogs and your cats can fall into this as well, so they can get these worms as well. And so what happens is they develop the worm, we release the proglottids into the environment, the prey eats the grass around that environment, and they end up getting the cysts. So it moves from the intestines into the body, and these are called hydatid cysts. Sounds like I'm stuttering. So hydatid cysts is just the name of the cyst structure that's created in the tissues. And then how does the predator get it? Eating the prey, right? He's obviously not cooking this on a grill. So he's gonna pick it back up. So, you know, whether it's a wolf or a cat, etc. We can get it by accident, typically because we come into contact, maybe camping or something, you touch something. It's not that easy, so don't get scared about that kind of stuff. It's not as easy to pick it up. But one of the things, okay, this is terrible. There are cases where people have gotten them from their dogs, right? So they can have these. And so if you have a dog that licks your face a lot, or your mouth, or, you know, see some crazy things on TV, you could potentially, because where else do they lick? Let's be honest, you know, they could potentially give it to you. Extremely rare. That is like probably, that's why it was in a medical journal, because it was such a rare instance of Echinococcus in humans because they picked it up from their dog that way. Yes.
So I listen to a lot of true crime like
Oh, boy.
podcasts.
Yeah.
And so this lady, this older lady, she fell at her house and she fell in her bathroom, and so she can, you know, took her a while to get a hold of family to come and get her. And the granddaughter finally was able to get over there and check her out, and she had like a gash in her leg. Well, the dog was trying to console. She brought her dog over to console her, and the dog was licking into the wound.
Okay.
And the woman actually died because of whatever the bacteria was on the dog's tongue, got into her and caused her
Into the deep
sepsis,
into the
and she
wound.
died.
It was opportunistic. Yeah.
Yeah.
I mean, these organisms do that. So why are we considered a dead-end host? Because we're typically not eaten by a predator, right? So we're not gonna, cat, you die maybe,
No more
or you're out
T-Rexes.
hiking, something like that. So we're not usually in their life cycle because we're not typically prey. So that's why we're considered a dead-end host for this one. Kind of like it was for, what is it called? Toxoplasmosis was that way as well, because we're not gonna get eaten by our cats most of the time. Yes?
You ever come across that video that supposed to be this dog? It was like in the news.
Okay.
The dog licked someone's face, and I don't know what the name of the organism was, but the person had to get all 4 appendages removed, like gone.
Oh, probably picked up staph of some kind, yeah, one of the
Yes.
scary ones.
But I don't know.
You don't think it's real?
No.
Yeah. Yeah, well, I mean, it just depends upon the host too. Like normal healthy people, that may not be an issue, but if you're immunocompromised, you are at higher risk for that kind of stuff. You can end up in more trouble.
But the appendages though?
Yeah,
Like gone?
oh yeah. We talked about some of the diseases like the Neisseria, gonorrhea, that one, or not gonorrhea, but meningitis, it can cause meningitis. And so you end up with blood clots. And that comes from a bacteria in your nose, so, right? So it can happen. They probably got something like that. Caused a toxin gets released, causes blood clotting to occur, and so you get DVTs in the different locations, and you can lose your limbs. It's terrible. Yeah. All right, that was fun. All right, so let's talk about a little sweet guy here. This is a planaria. I don't know if you can see it, but he has super goofy eyes. He does. so cute. I love planaria.
So funny.
It's very accurate when growing. This is his head.
He's kind of cross-eyed.
Yeah,
He's
he has these,
cross-eyed.
he has these things called eye spots. So he doesn't
Oh,
have true eyes, but he can detect light. And so it allows him to go towards the top of the pond or towards the bottom of the pond. So this is also a helmet. This is a trematode. But this is a completely free living organism that you find in ponds. They're about this big. Very, very fun. They can move around. But what's very interesting about planaria, I'll post a video, is all of their cells are stem, all right? So stem cells are unique in that they maintain access to all their genetic material. So if I cut my arm off, the cells that are remaining know how to reform that arm, right? That doesn't happen in us, but we see that in lizards, etc. So this guy, if I cut him here, this will regrow a new body and this will regrow a new head, because all of the cells have access to all of the genetic material. You can literally cut it into 7 pieces and you'll have 7 planaria when you're done. They do some messed up things. They cut them this way and then it ends up with 2 full heads as a result. That's the video I'm gonna post. So they're kind of fighting direction of where to go to the 2 heads. So then you gotta cut them and like restore it, you know, it's kind of messed up. But they're very interesting. So they're trying to understand, there's a lot of research that happens with planaria because they want to understand, why are they stem? Why are we able to access that information? Is there certain proteins involved, certain systems that we could make and then potentially apply to an area where you lost an arm and regrow it? You know, so there's a lot of interesting research that goes on with planaria. So it's not all horrible. They're not all developing cysts in your brain. Some of them are doing some very interesting things.
How big is that?
They're about maybe a centimeter in length. If you do pond water, you usually can find them. They're usually in pond water.
What if you grind it?
What do you grind it?
Yeah.
I don't think the stem would come to us because the genetics isn't quite the same, the factors that are in it. But if we could figure out what the factors are, we could put it into a plasmid, make a bacteria grow it for us, and then take it as a supplement or a drug of some kind. So it's kind of cool. Yeah. Yes, Naya.
They were saying that jellyfish are kind of like the same thing, but they can actually regenerate brain
They can.
cells.
Well, it's more like starfish.
The starfish?
do that, yeah.
And they were saying that, but they're not from this planet. That they came in
They
from
always
like
do
an
that, yeah,
asteroid
yeah.
somewhere.
They always do that. That's like tardigrades, water bears. Have you ever heard of tardigrades? They were in the Ant-Man movie, like this little thing with this face. They look like the Alice in Wonderland caterpillar. They look very much like that, except they have a face like a skull. It's scary. But they are everywhere and they're pretty regenerative as well. So these smaller organisms can do things that we cannot, which is interesting. And its mouth is actually underneath it. It's a tube that comes out, so that eats from the bottom. So I'll post videos so you can see it. All right, so let's talk about one more, and that's the nematodes. These are the roundworms. So these guys are cylindrical. Sometimes they'll have a tip, a little pointy tip, but they're not flat, like we see with our other group of cestodes and trematodes. So these are complete digestive systems. They're all dioecious. So these are the ones that are going to have both male and females, but usually there's either a groove they sit in or the spicules that holds on to the female. These can be free living or they can be parasitic. So we can find these as part of our systems. So the one, we're going to talk about a couple of them. One of them is ascariasis. This is the most common infection in the entire world in terms of nematodes. Very, very common. Humans are kind of the host. We're the host. It does require the embryo to go outside of the body, and then you can get reinfected. So notice there's no other organisms involved, but we have the male and the female worms. These are pretty decent size, usually between 15 and 30 centimeters. So they're pretty big. You can have multiples of these, so it's common to have less than 5, but depending upon how big of a dose of eggs that you ingested, you could have thousands of these worms inside of your intestinal tract. Again, surprisingly, it usually doesn't cause problems for most people, but this is one, if you have a large worm load, especially if you're a child, it can cause blockages. So they can have problems, a swollen belly, that kind of thing will end up happening. because of the blockage of the animal. So it's better to get rid of it in these cases. So this is living in your intestine. The males and females are generating, probably over the course of the lifetime of the female, maybe 200,000 eggs. So it's a constant life cycle. They live about 5 years in total. And so these are leave the body through fecal matter. It does have to develop outside of the body through the stages you see here, and you can reinfect yourself by doing contaminated food or water. So this is, again, fecal oral transmission. Again, very common in developing parts of the world. And then the last one we're going to talk about is one we see here, especially if you've had small children in daycare. Oh yeah, pinworms, right? So this is Enterobius. This organism can be very, very common in little kids because little kids are gross. If you want nothing else, they scratch and touch and scratch and touch and scratch and touch and scratch and touch, right? So they get stuff everywhere, and then they touch and put things in their mouths, and that's how we get it. This is a fecal oral transmission, once again. Very, very common. So don't feel bad if your kid gets pinworms. It happens, all right? It just does. We are animals, all right? These are pretty small, about 2 to 12 millimeters. So think like a millimeter is 1/10 of a centimeter. So maybe around that size, very, very small. You can have large numbers of these again, or you can have very small. It just depends upon how many eggs you ingest, because they don't reproduce inside of your body. It's actually outside of your body. Very simple. Look at this kid. Doing exactly what I said, all right? So you've got the male and the female inside. This tends to be in the lower intestine. And this is what they do is the female comes out at night and she actually lays her eggs around the anus. So it's on the outside of the anus where they lay the eggs. So you can imagine that's irritating. So kids will scratch at that and then they'll touch or they'll reinfect, right? So you can end up getting more and more pinworms because you're just touching and reinfecting. The way that you can find them is you take a tape, kind of to a tape pull right off the anus, and then you can look under the microscope and look for the presence of the eggs from that date. Or you can go based on age, symptoms, location, etc., or do PCR. Yes?
So, when I was in high school, super gross, but some friends share water bottles.
Oh yeah.
My almost entire high school got treated for pinworms.
Oh,
We
man.
have no idea where it started from, but it's a treat.
Oh, we know where it started from.
Specifically
Who
from,
it was,
but
yeah.
We had like a whole.
It was somebody's
lots of
little brother,
parents.
probably little sister
But
started
is
it.
it true that sharing a water bottle with somebody?
Yeah, it
you
would
can
be
get
in
it?
there. Why wouldn't it?
Yeah.
I mean, you gotta contaminate it though.
So, like, somebody has an irritation, whatever, touched their water bottle, their friend drinks it, can you still have eggs like throughout the
I
day?
don't know if it'd be on the mouth, cause your mouth is pretty
'Cause
inhospitable to a lot of
80%
organisms.
of my school had to get treated for pinworms. I was one of the 80%.
That got told you
It
that.
was not fun.
Yeah.
So, I mean,
It
it wasn't
probably,
anything crazy, you got
this
the
may have been one of the
location books.
locations, but it's more likely it was other places as well, cause if you touch other places, they touch it, you touch your mouth. So it wasn't directly the water that was doing it, cause it's more of a phonite transmission that does it than the water
Well,
necessarily.
no, like, I mean, like drinking off the same straw as somebody else.
Oh, yeah, yeah,
So like, you're sharing drinks, like drinking a soda can, and you're
Yeah.
like, okay.
yeah, gross. Okay, so these are some things to keep in mind when you're thinking about helmets. How do you classify them? How can you tell whether or not this is the organism you have? Definitely shape and size. How developed are their organs? Do they have hooks, suckers? What kind of reproduction? Is it monoecious? Is it dioecious that we're looking at? Who's the hosts? Who's the definitive hosts? And then oftentimes with these, this is again going to be fecal samples. You're gonna look at tape pulls. You're gonna do that kind of thing. And look under microscope to actually look for structure. Very common. Although it is possible to also do PCR, so you can look for the genetic material in an individual to see if it is present, because they do shed, right? So they're gonna shed some of those cells as well. All right, scary stuff. Yes, Janine?
So for the exam, because there's like a lot of organisms, would that be like something that you're basing your questions off, like the classification identification, or like what you would know for each?
So I would know what the name of the organism is for each. I would know the life cycle of some of these different organisms. I would know, one of the things I like to do is to give you an unknown and say, this has these characteristics. Give me a reason why you think it's a helmet or not a helmet, right? So kind of understanding the general broad. I'm not as interested in like CDC definitions of everything. I have to think about it a little bit more. Yeah. So the basics of what is, particularly like when we talked about how would I know if it was a fungus? What are the basic structures of a fungus? What are the basic structures of a helmet? Trematode versus a nematode versus a cestode, right? And then I might give you an example of a transmission cycle. What's the definitive, what's the intermediate host, right? So that kind of thing. More so than having to absolutely identify every worm and every, because you can always look that up, right? But I want you to know it's not a bacteria, right? Or it's not a virus. What's the defining features of those, okay? About a little bit more. All right, so we are gonna go ahead, yes, Gil, sorry.
Quick question.
Sure.
Is ringworm a parasite or is it a fungi?
It's a fungi. Yeah, so ringworm is a fungus. It's a dermatophyte. Yeah, so they call it ringworm because it kind of looks like a helmet under your skin, but it's a fungus. So it's just a subcutaneous. It's not subcutaneous. It's cutaneous fungus infection, mycosis. Just like it's like athlete's foot, basically similar. All right, so let's go ahead and get started on this. I will be posting the vast majority of this lecture, obviously, so you can take a look at that before class on Tuesday, so we can review some of the material that's found in here. We are going to be talking about viruses, viroids, and prions, all right? These are not alive, so they are all intercellular pathogens. They are reliant on the host to do most of the work because they're very simplified, so they don't typically do translation. They can do some transcription, as we'll see, particularly viruses have to do some transcription. But overall, they're completely reliant on the host. All right, so let's start with viruses. So this is the category, super fun. This is, kind of interesting. If you are alive on this planet, bacteria through human beings, you have a virus that attacks you. So every living organism has viruses. So everybody has this issue. Everyone's genetic material has evidence of viral infection. Isn't that crazy? So like part of our immune system actually has evidence of viral infection in it. So the things that were attacking us actually helped us develop the resistance to them, which is interesting. The placenta is thought to have developed through viral influence as well, which is crazy, right? So you can see the influence in our genetic material. These are obligatory intracellular parasites. They have very specific components. If I'm talking about a virus, a base virus has nucleic acid. This means it's either RNA or DNA. It's never both. It's one or the other. And it's going to have a protein coat. The protein coat is called a capsid. Not all viruses even have it, but the vast majority do. So the very simplest virus has the protein coat, and then inside it has a piece of nucleic acid. On top of this, they can have some other flourishes we'll see, but one of the key things, there is no ribosomes, there is no ATP generating mechanisms, they don't do cellular respiration, they don't do any type of translation. And because of that, as we'll talk later, they are resistant to antimicrobial drugs, specific for bacteria, right? So they're not gonna work. Some antivirals, we don't have a lot of antivirals, but there's some antivirals. So you're not gonna be able to take antibiotics in the typical sense to be able to fix it, all right? All right, so in addition to our nucleic acid and our protein coat, sometimes they have something called an envelope. So an envelope is a lipid layer on the outside. This is gonna sound creepy, but it's basically the skin of the host. So what they do is when they infect the cell, they then push through the plasma membrane on the way out and they take it with them. So it is like the skin of the host, right? So they end up taking components of the host with it. So some are enveloped and some are non-enveloped. So if they don't have it, they're known as being non-enveloped, all right? In addition, they can also have spikes. So spike proteins, these are generated by the virus, and it gives it its little alien appearance, right?
COVID.
Like COVID, yep, exactly. So it can have the spike proteins on the outside, it just depends upon the virus. As I said, some viruses are just the capsid and the nucleic acid. So the spikes, as well as the corners or the components of the capsid, help it to target its host, all right? So there's going to definitely be a host range. Host range is sad. The host range has to do with the ability of the virus to target a cell. So on the outside of the cell, there's these little receptors. It is not a virus-specific receptor. It's usually for something else the cell needs, like food, or vasoconstriction or vasodilation. It's a protein place. And so the virus has tricked the cell into recognizing it as something it is not. So it's basically the wolf in sheep's clothing. So this is a huge virus compared to myself. But it basically will trigger it to go, hey, this is friendly, bring me inside. So in order for that to happen, the host has to have the receptor that's recognized by the virus, right? So that's why, there's viruses that target yeast, there's viruses that target bacteria, and there's viruses that target humans, right? So we're not gonna get our dog's viruses necessarily unless it mutates, and now it can recognize our receptors, right? So that's the zoonosis that can occur, right? So you can change a little bit. All right, so these are the size comparison. Here is a yeast cell, so this is like our eukaryotic cell. You can see it's a budding yeast because it has the little mini-me coming off the side. Here's an E. coli, here's a streptococcus, and then you can see the rickettsia that we have here. And so we have viruses that can be very, very small. This is polio, adenovirus that causes gastrointestinal issues. Here's your influenza. And then we have some really huge ones. These are called megaviruses. That's my favorite, megaviruses. And they can be almost the same size as a bacteria in some cases. For some reason, they find them frequently in the sewage systems in Norway. I don't know what's going on over there, or they're hunting for them. But these are almost cell-like. So Pandora virus, which sounds very ominous, actually has rudimentary ribosomes. So it can almost do translation. So it almost feels like a cell that has decayed in function a little bit. It's like hard to tell if it went that way or it's going the other way, right? It's becoming something that we're not expecting. All right, so another term to kind of be used to is the word viron. So viron is a single virus, and these are examples, again, of that capsid with the nucleic acid. This one is not enveloped, and here we have it with the envelope and the spikes as well, all right? All right, so let's talk a little bit about the nucleic acid. It's a little different than what we've done. All right, so we have done DNA to DNA, right? We are DNA-based organisms. We can do DNA replication, correct? We can do DNA to RNA, right? What is this? Transcription, right? So this is the normal form for all living organisms. And then we can take that RNA and we can convert it to protein, right? So this is the gene expression component that we utilize the DNA for. So all living organisms are capable of these three processes. Thing we cannot do is RNA to RNA. No, we cannot do that, right? This is a type of transcription, but is not a transcription we're capable of as living people or living organisms. And then the other one, which freaks people out a lot, is RNA to DNA, right? We cannot do either one of these as living organisms. It's completely impossible. However, viruses can, right? So these are viral activities. Viruses have to bring with them enzymes to allow this to happen, but they are capable of RNA to RNA transcription, and they can do retro transcription, which is RNA to DNA. Okay, so these are the ones that they do that. So this means that their genome can be either DNA or RNA because they're capable of those two processes. So our DNA can come in some flavors. So DNA for us is always double-stranded. That's normal. So this is abbreviated DS for double-stranded. So double-stranded DNA is the normal function, correct? Viruses can be single-stranded DNA, right? That's why if we have single-stranded DNA in our bodies, remember, I don't remember in replication, we get the little telomeres that get shortened every time because we have to eliminate single strand because our bodies recognize that as bad. It's a virus that's very bad. So any single-stranded DNA is going to be removed and destroyed. In addition to this, we can also do RNA as a virus. We can do single-stranded. We can do double-stranded RNA. That's creepy, right? That's new. And this one here is unusual because we can have plus and we can have minus. So these are the two types that you can have. These are all the components that you can have as a virus. You can only have one, but you can be one of those. Yes?
For the RNA to DNA, I noticed you used the word retro, so that's a retrovirus.
Yes.
That's
So it's a retro, uses an enzyme called reverse transcriptase. So you see reverse transcription right in the name, right? So reverse transcriptase. So let's talk a little bit about plus and minus, all right? And then we'll stop. So plus is known as sense. Minus is known as antisense. Okay, so plus single-stranded RNA, it essentially looks like an mRNA, right? So it makes sense to the ribosome. So if that shows up inside of the cell from the poor little virus getting inside, it will immediately get translated, right? So this is a makes sense to the ribosome plus single-stranded RNA looks like mRNA, right? So that's coronaviruses, for instance. So SARS-CoV-2 is a plus strand, single-stranded RNA. That gets dropped inside of the cytoplasm. Our stupid little ribosome starts translating all of that and then takes over everything, right? Minus is antisense. What do you think that means?
It doesn't make sense.
Does not make sense. It is the wrong strand. So this one runs 5 to 3. This one runs 3 to 5. Probably thought those labels were never coming back, but there they are, right? So it is the wrong direction. So when this shows up on the scene, our ribosomes do not recognize it. So what has to happen immediately is it also has to provide an enzyme to do this. So it'll take that strand and actually copy it immediately into a plus sense strand. So then it gets translated, okay? All right, so I'm gonna go ahead and stop there. So look for the rest of the lecture online. Let me know if you have questions next Tuesday. And good luck with your grandstanding on Thursday. I'll be in Salt Lake. Yay! It's gonna be good. I'm excited. I'm just not a person that likes to
I'm
do my
sorry.
routines. I'm like,
I'm sorry you'll be in Salt Lake. I lived in Salt Lake for most of my, when I was a kid in the 80s. My brother was born there and he disowned it.